already covered what is translation in the previous class. So translation is the process through which there is the protein which is synthesized using mRNA. So this mRNA transforms into tRNA by the process of activation, and then there is a phase of initiation, elongation and termination. The Sheila-Diglario sequence is not required in the case of eukaryotes, and in eukaryotes there is an 80S ribosomal unit divided into 60S and 40S which is there, and the 4-letter nucleic acid sequence changes into a 20-letter protein sequence series. So first stage is the activation of amino acid, which is common for both, and this is where the tRNA picks up most of the amino acids. So the amino acid combines with ATP and enzyme to form the amino acyl adenylate enzyme complex. This combines with tRNA to form a charged tRNA plus AMP and energy, and this is very important, the 5-cap series to the 3 end. So 5' to 3 end is the movement of the eukaryotic mRNA sequence. Now in the second stage, what is important is there are 9 initiation factors. So these are the initiation factors that are given here. Now these initiation factors are important. The first stage is the formation of a pre-initiation complex which consists of the 40S unit. Now once the 40S subunit combines with these given initiation factors, this is known as the pre-initiation complex, and then you have the IEFA release which takes place, and the complex moves around the 3' sequence, and finally there is a movement from 5' to 3', and a complex position over the initiation codon is seen, and the 60S ribosomal unit is actually binding with the 40S unit to form the 80S microsomal unit. The next is the process of elongation. The elongation in eukaryotes is very similar to prokaryotes. However, it requires 3 elongation factors. The elongation factors are EF1A, EF1BG and EF2. Okay, and their counterparts in the prokaryotes are EFTU, EFTS and EFG. Clear? Also, as eukaryote ribosome do not have E site, the uncharged tRNA are expelled directly from the P site. So again, a very important thing that the eukaryote ribosomes do not have a E site. So any kind of unexpelled ribosomes, the uncharged tRNA are expelled from the P site, and the elongation of the peptide chain takes place. So here is how the elongation takes place, and the polypeptide lives in the grooves of the larger subunit. And finally is the stage of termination. The stage of termination in the case of eukaryote is again similar to the prokaryotes. There are stop codons with the UAG, UAA and UGA. The 3 stop codons which are there are important. Their factors, which is RF1 and RF2, are important. In prokaryotes, in eukaryotes it's RF1 and RF2. In eukaryotes it is single ERF1 which recognizes all the 3 termination codons, and with the help of EF3 the ribosomal subunits are released, and this prevents the release or the reassociation of the subunits in absence of the initiation factors. So the units get separated out. So here we understand how the separation takes place. Now here we have an example of the prokaryote, which is 50S and 30S. However, in eukaryotes it is the 60S and the 40S units which are taken into account. So this is a basic step of translation in eukaryotes. Prokaryote translation we have covered in a separate lecture, but this was just an overview to give you an idea about what is the process of translation and transcription in prokaryotes and eukaryotes. So wish you good luck, and in case you have any queries or doubts, do follow on the links below.